called the proton oligopeptide transporter, abbreviated PEPT, located in the apical membrane, intestine, epithelial cells, and the epithelial cells are the ones behind the side intestine that where the food grew. Also found in anal proximal tubule. What it does is coupling the downhill movement of one proton to the uphill movement of one dipeptide or one tripeptide. Interestingly, either an dipeptide or a tripeptide that is moved in response to the proton movement. In addition, PEPT also serves as the entry route for peptidomimetic drugs such as beta-lactam antibiotics. So it's very important in the movement of antibiotics of specific classes. So stomach emptying of acidic chyme into the early regions of the small intestine will create an acidic environment with a favorable transmembrane proton. Protons are the ones which determine how acidic the environment is. So the gradient caused by that acidity is what is responsible for PEPT-mediated transport of either di or tripeptides. PEPT is also thought, this is still under investigation, so maybe by the time you graduate, who knows, we might have definitive data, but PEPT is thought to be the dominant player in nitrogen absorption in the small intestine, and nitrogen absorption. So it has quite a number of functions, specifically movement of antibiotics, tripeptides, and also nitrogen, three key molecules. Sodium coupled bicarbonate transporters have other features which may need to be brought to your attention. They perform important acid-base balance. You know that bicarbonate ions are very important in balancing the pH. Both electroneutral, which are called NBCN, and electrogenic, which are NBCE, isoforms have been identified. I suspect you know what isoforms are. Isoforms? What's an isoform of a protein? What's an isoform of a protein? Anybody? Or shall I call on people? No, we are tired. It's late in the day. Oma. Oma? What's an isoform? Oma, Trevor, Granton, in case there's more than one. Yeah? Anybody?
Hello, Prof.
Yes, ma'am.
Let me try.
Yes, go ahead.
So isoform proteins are proteins which are similar and they perform a similar role.
Very good. Why would we have similar proteins which perform similar roles?
That one I'm not sure, but I just know isoforms are similar proteins that perform similar roles.
Your answer is here. This is a bicarbonate transporter, right?
Yeah.
It is balanced, but one is for electroneutral and the other one is for electrogenic. Okay? This is a very good example of form. All right? So they are both similar. The bicarbonate co-transporter, the protein structure is similar, but The similarity comes in the function of these two. The structure is similar. One is just classified as the electroneutral, and the other one is for electrogenic. So remarkably, NBCs belong to the same family as the chloride bicarbonate exchangers. So, exchangers and antiporters, what's the difference? Exchangers are also known as antiporters. They are found in many different cells and tissues, and they also perform a variety of very important physiological functions. Exchangers are sodium, calcium, protons, chloride, and bicarbonate. So I would like for you to have a very, very clear knowledge of what exchangers are, and please do not ever mistake them. Okay? So, the sodium calcium exchanger, known as NCX. If you note, we've moved from co-transporters to exchangers. And by the name, it's pretty much exchanging one or the other one. So the sodium carbon exchanger, abbreviated NCX, is ubiquitously found in many cells. Now, this is an important word for any biologist. So if you have ubiquitous, means expressed a lot, all over, not just limited. It's ubiquitously found in many cells and tissues, and plays an important role in the cytoplasmic calcium homeostasis. Remember, we've already met a very important role for calcium in release of vesicular contents, which have come from the Golgi apparatus. So the balance of the amount of calcium is so important that it needs a homeostatic control. And this function is performed by the sodium calcium exchanger. So what does it do? It movement of 3 sodium ions down the electrochemical gradient to the uphill movement, meaning that it's moving from a low to a high concentration of 1 calcium ion against a very electrochemical gradient. Remember that resting calcium concentration values in the cytoplasm and extra are 70 nanomolar to 2 molar respectively. So you can see the big change in the extracellular fluid. And obviously for that reason, NCX is electrogenic. I'm going to give you a second to take this down so you can chew on it as you take it down. If you have any questions on the sodium calcium exchanger, make sure you understand it before we move on. ubiquitously found exchanger that plays a key role in the regulation of cytoplasmic pH is the sodium hydrogen ion exchanger. This is a no-brainer because, as I said earlier, protons are very important in pH control. Here again, the sodium electrochemical gradient is utilized by the exchanger to extrude protons from the cell with a one-to-one coupling stoichiometry ratio. Therefore, the sodium hydrogen exchanger is classified as electroneutral. Then there's a chloride bicarbonate exchanger. This is widely distributed, obviously, because the pH control is very important. So chloride is also referred to as the anion exchanger, or simply AE. This is the only one with 2 letters. The coupling stoichiometry of transport is 1 chloride to 1 bicarbonate, and therefore the exchanger is electroneutral. These exchangers are special because they belong to the same family as the sodium bicarbonate cotransporters. The direction of transport by the chloride bicarbonate exchanger is governed by the concentration gradients of both chloride ions and bicarbonate ions. I bet you begin to see a pattern here. So it's kind of repetitive, but it's important to highlight these things. So here's an example. In red blood cells, also biologically known as erythrocytes, traveling in systemic capillaries. You do know why these capillaries are called systemic. That's in your physiology. Excess production of bicarbonate in the cytoplasm of erythrocytes as a result of carbon dioxide hydration reaction, which is catalyzed by carbonic anhydrase, will drive the outward translocation of bicarbonate ions in exchange for inward translocation of chloride ions. So when the red cells pass through lung capillaries, for example, high oxygen, the direction of the transport is reversed such that the inward translocation of bicarbonate is coupled to outward translocation of chloride. The bicarbonate that enters the cells is ultimately gives rise to carbon dioxide catalyzed by carbonic anhydrase, which then leaves the cell and ultimately the capillaries to enter air-filled alveoli of lungs, where it is then exhaled into the atmosphere. So basically, we've taken the chloride bicarbonate exchanger, taken the red blood cells in systemic circulation, moved through to the lungs, included carbonic anhydrase processing of carbon dioxide, and pretty much exhaled it into the atmosphere. So we have looked at how molecules are involved in removal of carbon dioxide from circulation, from the systemic through to the alveoli. I'll let you copy this example because this is the whole point that we're talking about exchanges and transport. Stephen Boy Wangui, your hand is raised. You have a question? Stephen? Your hand is raised. No? Okay. So let us look at 3 examples of these exchanges again. The sodium calcium exchanger, NCX, 3 sodiums exchanged for 1 calcium. The coupling stoichiometry is 3:1 ratio. The sodium proton or hydrogen exchanger, 1 sodium moving in to every proton that goes out. Coupling stoichiometry again, 1:1. And then the chloride bicarbonate exchanger, the only one with 2 letters, AE, 1 chloride to 1 bicarbonate. So you're literally exchanging 1 molecule for the other. So many transporters have multiple isoforms and features such as tissue localization, coupling stoichiometry, and also substrate specificity. And this may vary with the different isoforms. I've just given you a few examples of some and also shown you how co-transporters differ from exchangers. Moreover, there could be differences among ortholog transporters, and orthologs, of course, as you probably know, is species differences such as humans, rats, and mouse. Why is this important? Many of you might end up in research and you would need to know that the orthologs are differences in the same thing which would be found in the human or the rat or the mouse. So don't go out there and appear like we never taught you. So we've come to the end of secondary active transport. I think I've given you quite a bit. You have a lot of resources where you can go and read up some more. And my view is that you understand the principles and the differences in some of these molecules and how they are involved in some of the physiology are focused on red blood cells in systemic versus lung exchange of carbon dioxide. If you noted in there, I threw in a side issue of the carbonic anhydrase, which is probably something you covered in enzymology. I've also thrown up a lot of examples in pancreatic acinar cells. I've thrown in examples in the kidney. And so these, and also intestines. So these are things which occur in tissue system throughout the body, and these exchanges might, and exchanges and co-transporters are key elements of on the lipid membrane bilayer and those integral proteins. Any questions while I load up the next? Actually, what we were supposed to do for this lecture. Any questions? No? Are you just tired? Are you? Can you hear me? Can you hear me, or what's going on?
Yes, we can hear you. Yes, we can hear you, Prof.
Mike.
Yes.
What's wrong with your cousins? Sorry?
No problem. There's no problem. There's no problem. Prof, yeah, it's a bit tired, but moving out.
But you're the ones who told me you're okay for this time slot.
Yes, we are okay with it.
Okay, Mildred, thank you. Phoebe, thank you. Okay. Yeah. I'm the one who should be tired. We were examining a PhD most of the morning. Okay, so I'll take you back to what we did before. Transport through channels is very fast. Okay. You have the closed formation and moving materials through, and then when the channels open, you get the materials coming through. The flow rate is approximately 1,000 times greater than the transport would be by carrier proteins, the exchanges and the co-transporters we've talked about. So the channels really allow things to move much, much faster. This is just kind of to remind you of something we've done because we'll be touching on it again. And ion channels are very selective. The narrow pores of channels will restrict passage of the appropriate size and charge. We've been through that. And of course, the specific channel proteins allow the passage of sodium, potassium, calcium, and chloride across the membrane. You can see some repetition in what we've just gone through on the co-transporters and the exchanges. Ion channels are not permanently open. They are regulated, of course, by gates, and these gates transiently open in response to specific stimuli. We've got the ligand-gated channels, which must have binding of a ligand to a receptor or other signaling molecules which cause these channels to open. The others are voltage-gated. We've been through those, the electrochemical. Voltage-gated channels open in response to changes in electric potential across the plasma membrane. And we've talked about what this electric potential is supposed to look like. So the fundamental role of ion channels in the transmission of electric impulse in nerve cells is very important. I bring this up again because we'll be doing this in detail. We talked about the sodium potassium pump and the requirement for ATP in energy. So it is known as the sodium potassium ATPase pump. And then we talked about the channel proteins, which allow free flow of potassium channels depending on the need, either intracellularly or extracellularly. And of course, the concentration gradient as well as the electrochemical gradient will be necessary depending on the physiological function of that particular cell and that particular time. So we've gone through the fact that ion channels are not permanently open. Ion channels, ion pumps, and some antiporters will transport only ions. In all cases, the rate and extent of transport across the membrane is influenced not only by the concentrations of the two sides of the membrane, but also by the voltage that exists, across the membrane. In addition, of course, to ion pumps, which transport ions against their gradient, the concentration gradient, I beg your pardon, the plasma membrane contains channel proteins that allow principal cellular ions, sodium, potassium, calcium, and chloride. All those co-transport and exchange mechanisms we've just gone through to move through different rates down the concentration gradient. Ion concentration gradients and selective movements of ions through channels will create a difference in the voltage across the plasma membrane. So we're just putting it together with what we had done before. Now, let us look at ionic gradients and electric potential maintenance across the plasma membrane. Now we are going to focus and include the electric potential and how these ion gradients that we have created through transportation and exchanges happens. So in cells, the ionic composition of the cytosol is usually different from that of the surrounding fluid. The extracellular fluid can be in the lumen or in the extracellular matrix. In virtually all cells, including microbial, plant, and animal cells, cytosolic pH is kept near pH 7.2. And we've just gone through how this is regulated through the bicarbonate transport. The cytosolic concentration of potassium is much higher than that of sodium. Those are key elements that we went into detail earlier on. So this is very specific about cellular ion composition, and it is an important thing to keep in mind as we go along. So in typical cells and blood, concentration of vertebrates, of cellular and blood molecules of potassium, sodium, chloride, bicarbonate, magnesium, and calcium vary. We talked earlier on about sodium, 45 in blood, and inside the cell it is 12. In potassium, 4 in blood, and 139 in blood. And this is really important to have this difference in the ionic concentration, because the regulation will be performed by the exchanges and the co-transporter. So both vertebrates and invertebrates, in other words, living, breathing things, the concentration of potassium is 20 to 40 times higher in the cell than in the blood. The concentration of sodium is 8 to 12 times lower in cells than in the blood. And the concentration of calcium free in the cytosol is less than 0.2 micromolar, 1,000 or more times lower than in the blood. In here, you will notice that we are talking about free calcium. Why are we talking about free calcium? Those who took cell biology with me remember that in cells, there is a protein which binds calcium called calmodulin. C-A-L, modulin. I'll type it here. I keep forgetting to put it in the slides. Calmodulin is very important because the CAL is calcium and modulin is modulating. So basically it is a protein that modulates how much free calcium there is in the cell. Why do you need such a protein? It's very important because this, as we talked about, is very important intracellularly and needs to be available at the drop of a hat. So calmodulin will release any calcium that is needed, and once that reaction is finished, it will bind any excess calcium so that you don't have a lot of free floating calcium inside the cell. I'll let you copy this down because it's very important to have this background as we go along, because now we are going to look at specific examples of ionic balance and the physiology of that ionic balance in different situations in human physiological systems. So I'll let you copy this down. 2 minutes. Ionic gradients and electric potential of a membrane drive many biological processes. For example, when you have opening and closing of sodium, potassium, and these are essential to the conduction of electric impulse down an axon of a nerve cell. And I'll be using this example a lot. This is where you find many of the complicated movements. So a membrane electric potential, sodium concentration are essential. In many animal cells, sodium concentration gradient and the membrane electric potential power the concentration gradient. This transport is catalyzed by ion-linked symport and antiport proteins, co-transporters, and co-exchanges. In most cells, a rise in the cytosolic calcium concentration is an important regulatory signal. And this regulatory signal is what is responsible for initiating contraction in muscle cells and triggering secretion of digestive enzymes in the exocrine pancreatic cells. So those tissues and organs that literally have innervation to cause movement or deposition of other molecules. So let us go to the lab for a minute and look at an experimental example here. We have a beaker. This is a rough experiment. We have a beaker with one side representing the cell cytosol. And how do we know it is the cytosol? Because we have put in the concentration of sodium chloride and potassium chloride that we know mimics what you find in the cytosol. Then you have the extracellular medium, which now in this beaker represents the extracellular fluid. And in this case, you have the concentration of sodium chloride. You can see it is higher than intracellular, 10 times, 150 millimolar to 15 millimolar, and also potassium chloride, which is lower in the extracellular compartment than in the intracellular compartment. Remember what we were talking about, potassium ions, where they're highest. Do you remember that? Then you have electrodes which are connected to a potentiometer. And here you have zero potential difference over here on the potentiometer. And we agreed that at minus 75 millivolts is the resting of the cell. So here we've put minus 60 because it varies. It's not cast on stone that it is minus 75. It could range anywhere from minus 50 all the way to minus 90. And then here is plus 60. So sodium ions tend to move down their concentration gradient from the right side to the left. When they move, they will leave an excess of chloride ions, which are negatively charged compared to the sodium ions on the right side. This will generate positive sodium ions compared with chloride ions on the left side over here. So I will let you copy this experimental example because we are going to take it to completion to see how these charges align on the membrane here. Okay, so I'll let you copy this as we go along. We started off with 30, we are down to 28. There are people who've taken off from class. Okay, I'll just copy. Yeah, just copied the attendance. Right. So the excess of sodium to the left and excess of chloride, which is left here when sodium moved across, remain near the respective surfaces on the membrane alongside here, since it is a capacitator. The excess positive charges on one side of the membrane are attracted to the negative charges on the other side. So on this side here, I hope you can see the red dot, will be the excess chloride which have remained after sodium moved across the membrane over onto this side. So obviously, since they are opposite, the positive will remain attracted to the negative charges. So the resulting separation of these charges is then measured by the potentiometer as an electric potential or voltage, with the right side of the membrane having excess negative charge with respect to the positive on the left side of the membrane. So you can see here the sodium's moving across, the chlorides remaining, they're negatively charged. So you have positives and negatives, and we had an example of this earlier, I think, in yesterday's lecture, where the positives remain by the membrane and as the internal in the cytosolic membrane, and then the positives, the negatives on the extracellular face, or vice versa, depending on what movement we have observed across the membrane. Obviously, the sodium moving across is not just going to go through the membrane at random. It will have to go through what we have already come across as the sodium channels. So when you are explaining such a thing, you don't leave out the fact that this is going through the sodium channel, which we looked at length in very early lectures. So I don't need to go through exactly how that looks. So as more and more sodium ions move through the channels, these channels across the membrane, the magnitude of this charge difference, which is the voltage, increases. However, if you have continued right to left movement, the movement of sodium ions is eventually inhibited by the mutual repulsion between the excess positive here and positive charges which have been accumulated on the cytosolic side of the membrane by the attraction of sodium ions to the excess negative charge in here. So this is going to inhibit by attracting sodium ions, so that the sodium ions don't move across the channels into the cytosolic compartment. So this system will eventually reach equilibrium, and at equilibrium, the two opposing factors that determined the movement of sodium ions, that is the membrane electric potential and the ion concentration gradient, which we started off with, balance each other out. And that at that point, we will have reached equilibrium potential. So you can copy this final or second very important experimental explanation. And it is at this point that equilibrium has been accomplished at minus 59 to 60 volts in this particular example, with the right side negative with respect to the left. Are we together? Have I lost anybody? Fantastic. You guys are good. Have you done this read something before? I'm wondering why you're so easily malleable. So far, so good. Thank you, Vivian. Then I think online lectures are better than face-to-face. Who prefers online? Who prefers face-to-face? We've come a long way. If online is preferable. I thought you were kicking and screaming last semester. Okay. You are repulsed. You just had to jump in feet first. Just for future reference, if somebody as old as me is going to dive in and do this technology manenos and manage on my own, then you guys have no excuse. All right. So basically, this is just the same slide, but I had made it larger for scenarios where we are having physical class. So potassium channels, ion pumps, and the potassium concentration gradient is really important, as you've come now to appreciate. The concentration gradient drives the flow of ions through resting potassium channels, and this is generated by an ion pump that transports potassium ions into the cytosol from the extracellular medium, and the same channel drives potassium ions out. In the absence of this pump, I would ask you right now to type for me what this pump is called. The potassium concentration gradient could not be maintained, and eventually the membrane potential would fall. So the pump is absolutely essential for maintenance of the membrane potential. Things don't just float in and out. In addition to maintaining the membrane potential, you also have the requirement of the sodium potassium pump to maintain this integrity. So resting potassium channels show that the channel protein is built of 4 identical subunits. They look like so. This is just a rough sketch. And each subunit contains 2 membrane spanning alpha helices. So in total, there's 6, the 4 subunits, and then the membrane spanning alpha helices. And these partially line the ion conducting pore in the middle of the protein. The shorter looped P segment in here acts as a filter. I would rather say as a sensor rather than filter. And this sensor is the one that allows potassium, but not other ions, to enter the pore and cross the membrane. So there's a sensor here which picks only potassium and not other ions to pass through. So this is in the reference literature that I've given you to refer to, so you don't need to copy it, it will be in your books. How does this look like? This is an integral membrane protein that has been inserted as a transmembrane protein. In other words, it traverses the lipid bilayer, subunit 1, subunit 2, 3, all the way to subunit 6. And these are the transmembrane alpha helices. It has an amino terminal and a carboxy terminal, which are both within the intracellular space of the cell. And then, of course, here is the P-loop selectivity, ion selectivity filter that I just spoke to you about. And it is in here that it will select potassium and not any other ion to pass through. So we have finished the key elements of combining the carrier proteins, the channels, the integral membrane proteins, the co-transporters, the exchangers, and the physiological relevance of all these molecules that we've been talking about, as well as structures. We've talked about how they come from ribosomes on the endoplasmic reticulum through the Golgi apparatus, how they are transported in vesicles, and how they move out of the cell if they are for secretory proteins, and how they're inserted into the membrane, as in the case of this particular protein we're looking at in this slide. We've also talked about how bulk materials are moved from outside the cell through invagination of the plasma membrane, the formation of the basket, like clathrin-coated basket, and cleaving off of the vesicle. That vesicle goes and attaches to the early endosome, and in the early endosome you have the hydrolase enzymes which break off what is needed, and what is not needed is digested and killed. We have to remember that also cellular debris for materials which have died inside the cell and have been replaced are also cleared out in the lysosomes. So the early endosome fuses with the lysosome, and in here you get a vesicle pinching out, and when the vesicle pinches out, it also recycles the proteins which are necessary, such as the receptors, which are channeled back in vesicles to the plasma membrane, and by the process of exocytosis, binding of that vesicle to the lipid bilayer plasma membrane, they are reinserted there and reused. So if you have any questions, please let me know. Are we good? Is it fun? Is it crazy? Is it boring? Can I have some feedback? We're pretty much done for today, and I think you've got quite a bit to chew on today, and we've caught up gladly. You'll be happy to know. You're deathly quiet. The silence is deafening. Okay, let's go up to that point. You don't know what's coming. Good? Okay. Jotian. That's a lovely name. Okay, good. Evelyn Ruben, Darren Wamunga. Are you present or absent? Mildred Jayo. Mildred. So far so good, Evelyn. Cheryl Okwako. Cheryl.
Yeah. Aja tawin di five na tawin aja. Yeah.
Okay. Get Kamoy.
Hey.
Present.
Okay, good. Did you understand, Kip Kemoy? Or you're just present and dazed? Odhiambo Brian, you're awfully quiet. Okay, so I've typed in, please note the supplementary reading references in the course outline. Make sure you go and look at those. We'll pick up next week on Tuesday. Schedule is going bonkers. Okay, on Tuesday and Wednesday as usual. I think we've caught up, so we'll continue with a similar pace. So there isn't so much content in this particular course unit. There's just a little information, and then we'll go from there. Odhiambo has come back from his walk. I can see him typing. Any question? Mike, Odhiambo, go and read your course outline. If you have not and then you will ask me when the assignment is due. It is on the course outline.